Our second speaker today is Anna Calinawan from Mount Sinai in USA, and Anna is going to be talking about the
CPTAC data browser.
Hand over to you.
Hi, yeah. I'll be talking about the CPTAC data browser. A lot of the talks today have been mostly about APIs and sort of physical hardware and infrastructure. This is kind of more about front-end gains, specifically using progressive JavaScript frameworks. So thank you for having me. I'll be presenting about the CPTAC data browser, proteomics data visualization from the Clinical Proteomic Tumor Analysis Consortium. I'm a little bit sick, so please bear with me. So first things first, who is the CPTAC and CPTAC data browser? The National Cancer Institute in the US has started this Clinical Proteomic Tumor Analysis Consortium, and it's a national effort to accelerate the understanding of the molecular basis of cancer through proteogenomics specifically. So it starts with tissue source sites where they collect tissues and clinical data, goes on to proteomic characterization centers, off to the proteomic data analysis centers, off to translational research centers, and ultimately the cleaned up data should be going to the research community, and that's what I'm talking about today, is how do we present this data in a meaningful way to the research community. This unique big data resource is now in its third iteration, and we have generated and analyzed whole exome, whole genome, transcriptome, DNA methylome profiles, whole proteome, phosphoproteome, and mass spec profiles for about 1,000 patients for 6 solid tumor types. The raw data is available at the Proteomics Data Commons. It's not what I'll be talking about today, but it's a really great, if you are somebody who's interested in programmatically accessing raw data, it has a great API, has all these available tumor types from the last 3 iterations of CPTAC. So now we're going to get to the actual data browser and the problems that it solves. So with great data come great figures, and with great figures come great limitations. Let's say you have a paper and you get a heat map on that paper figure. At the end of the day, it can be very hard to customize or get deeper into that particular visualization, and all you're getting is the summary that's provided to you from the paper author. Here comes the CPTAC data browser, which is transitioning our data from static to dynamic visualizations. What does that mean? For example, we'll start with, let's say you get this figure where you have a static gene set. And this means that out of the entire data set that's been generated, here in particular we have mutation data, methylation data, copy number variations, proteome, phospho, RNA-seq. Of all of that data, of all of the genes, we have over like 20,000 genes, you will only get the ones that the author has decided to feature and focus on. What if, say, for example, you wanted to look at the proto-oncogene CRK in this context? I think the impulse would be to download the data and to write your own code to produce your own heat map, which kind of sucks if you don't actually write your own code, if you don't know how to code, or if you just don't want to do that. So now what we have instead is we can allow you to use the user-defined gene set, where basically I'm going to do exactly that case. You have, let's say, for example, the genes that are automatically on there. We add the gene of interest, CRK. It's going to take a little bit of time to load, so okay, there we go. And then now you can see that we have that track added. And this just allows you to have a more control over what you decide to see and what you're interested in beyond what was just given to you. Admittedly, this is something that already exists. Even back in CPTAC 2.0, back in 2016, you could allow for a specified user gene input. However, the patterns were static. So basically, the sample order is static, meaning if you wanted to explore deeper into specific patterns and ordering of things, you would have to still, once again, download the data and code your own, where you would reorder the samples and create your own heat map. This new visualizer allows you to sort by track, and that creates a much more dynamic thing. So let's say, for example, I'm particularly interested in chromosomal translocation events. I would simply take this track and sort it by ascending. If I wanted to look at the relationship of, if I wanted to look at 3-5 chromosomal translocation events, and I wanted to look at its relationship to 3-2 chromosomal translocation events, I would also sort that. And that allows me to see, oh, look, here's a pattern I wouldn't have seen otherwise, which is that they're essentially mutually exclusive, and I have just one sample that's a little bit, that has both cases. So it just allows for deeper biological insights if you have more reactivity and more options to get deeper into the figure. And then, of course, you have this option of filtering tracks. Essentially, this is creating a lot of noise if you're not particularly interested in that. For example, if you're only interested in the relationship of protein to phospho, the rest of it is detracting rather than providing information. So you can just click and remove tracks as you would like to. And that's just sort of a nicer way to visualize your data. And then finally, you can dig deeper if you have reactivity. So for example, you could get information about the genes. It's kind of a one-stop shop. That's the nice thing about a website versus like an image on a piece of paper. You can get information about the genes. Am I clicking around here? Okay, great. If we go back to this case and we can look at the errant sample, you can click on the sample, find out which sample it actually is. That's something that would be much harder to do with just a static piece of paper. We can view the histological images, specifically the tumor tissue organized by type. It's organized by, and you can see normal tissue, and it links to all of the CPTAC cancer images. And we can see the clinical data to see what's up with this particular sample. Is there something special about it that is different? Okay. And so again, this is just a way of getting deeper into your data without necessarily having to download the entire data set, parse it, process it, et cetera. Okay, and then here's a little bit of technology for technology track. This is actually a little bit of a digression, but since I wanted to talk specifically about front-end code for a little bit. Basically, progressive JavaScript frameworks are this amazing thing. They were mentioned this morning. Somebody mentioned React, I think. And so, as I'm sure many of you know, basically a website is an HTML document with styling CSS, and the primary thing that makes it interactive is your JavaScript. Traditionally, JavaScript, or like historically, JavaScript could be applied in a really very messy fashion, and that is a frustrating and difficult thing to deal with. Progressive JavaScript frameworks address a lot of these issues by allowing you to have components, and each component essentially has its own HTML, CSS, and JavaScript associated with it. So this is much more organized and also means you can update your front end in a much more modular way. The big players in progressive JavaScript frameworks are, as mentioned this morning, React, which comes from Facebook, Angular, which comes from Google, and Vue.js, which is my favorite, that comes from this guy, Evan You. And Vue.js is just more organized than Angular, I would say, and has a really nice ecosystem, including Apex charts, which is what I use to create these heat maps. I've actually written a tutorial, feel free to visit it, where it walks through Vue.js reactivity, how to install heat maps, how to customize them, and how to include them on your web page. So by all means, please feel free to visit that. And so what's next from the CPTAC Consortium with respect to data and visualization? The newest data releases are pediatric brain cancer and lung adenocarcinoma studies. Pediatric brain cancer is proteomic and phosphoproteomic analyses of 7 pediatric and adolescent and young adult histologies, including low-grade glioma, high-grade glioma, ependymoma, ganglioglioma, craniopharyngioma, atypical teratoid rhabdoid tumor, and medulloblastoma. And so the next question is, these are being uploaded to the Proteomics Data Commons now, but the next question is, how do you deal with something like, say, these 7 different tumor types when you have one large figure? This is another thing that we're doing. We're developing this next portal where you have the same functionality of being able to sort by like a specific gene and proteome, like by specific protein abundance of a specific gene, but then you can also get sort of more fine-tuned looks by organizing by diagnosis, and then the sorting is maintained, and so this allows you to have both a coarse and a more granular way of looking at the data. Okay. I would just like to thank everybody here at ISMB and also everyone at Mount Sinai who helped, and the officers at the Cancer Clinical Proteomics Research Office, and the members of the NCI CPTAC Consortium. This is a very small part of a very large effort. The APIs consumed were clinical data from the Proteomics Data Commons, images from the Cancer Imaging Archive, and gene information from NCBI. And then here are some helpful links. If you're interested specifically in kidney cancer, please visit ccrcc.cptacdataview.org, the Proteomics Data Commons, and the CPTAC Data Portal. Sign up for the listserv if you would like more information about the upcoming data dumps. Thank you.
So thank you very much. You covered a lot of things in a very short amount of time. Can I invite comments, questions, thoughts, observations? Start question. What?
Yeah, exactly. Yeah, I used Webpack. I used single file components and Vuex architecture. So I have like a single source of truth, and then just, yeah, lots of single file components. The tutorial that I mentioned shows you how to do it with a single HTML file, just to like make your life a little bit easier so you don't have to deal with this issue of like, yeah, configuration and compilation.
Okay. We've got time for questions.
Great presentation. So I just want to ask, like, all the data is available from this Proteomic data commons, the metadata. Is those metadata available through like some API access?
Yeah, so yes, Proteomic Data Commons has an API, and then the sort of like for the raw data, and then we are developing sort of the next level API for the metadata.
Oh, okay. This
Yeah.
is like the visualization portal sit on top of the.
Exactly. Yeah, exactly. Yeah.
Okay, so let's go off the image. Sorry.
Switch the lights there. Oh, the lights are off.
The lights could be on. Okay. Well, I think what we'll do is we'll say thanks so much to Anna once again, and just while we solve the sort of